DNA fingerprinting, also called DNA profiling, is a laboratory procedure that identifies the unique genetic makeup of an individual. The process focuses on variable number tandem repeats, VNTRs, or other polymorphic regions in the genome, particularly in the non-coding parts of DNA. Although these regions do not code for proteins, they exhibit a high degree of variation from one person to another. Non-coding DNA, a major portion of the human genome, does not directly code for proteins. These regions often have repetitive sequences. VNTRs and STRs, variable number tandem repeats, VNTRs, and short tandem repeats, STRs, are examples of repeated sequences that differ among individuals. Polymorphism. Because the length and number of repeats vary widely between people, these regions form the basis of fingerprinting, DNA fingerprinting steps. DNA isolation. Scientists collect a sample, example from blood, hair follicles, saliva. The cells are broken open, and DNA is carefully extracted and purified. Special proteins known as restriction endonucleases are used to cut the DNA at specific sequences. This creates fragments of varying lengths based on each individual's unique repeat patterns. The DNA fragments are placed in a gel and subjected to an electric field. Smaller fragments move faster through the gel, thus separating DNA fragments according to size. The separated fragments from the gel are transferred, blotted, onto a nylon or nitrocellulose membrane. This makes the DNA stable and accessible for further analysis. Hybridization with labeled probes. Specific VNTR or STR probes, often radioactively or fluorescently labeled, bind to complementary sequences on the DNA fragments. These probes highlight the areas of interest that display polymorphism. Autoradiography, or imaging. An X-ray film or other imaging technique is used to detect the labeled probes. The result is a series of distinct bands unique to each individual, known as the DNA fingerprint. Application. The application of DNA fingerprinting is extensive, owing to the technique's ability to distinguish individuals with remarkable precision. Some of the most notable uses include forensic investigations. Any DNA sample found at a crime scene, hair, blood, saliva, is profiled. Suspects' DNA can be compared against the crime scene profile to identify or eliminate individuals. Paternity and maternity testing. DNA fingerprinting can confirm biological relationships with high accuracy. This is particularly useful when determining the parents of an unclaimed infant or resolving family disputes. Population genetics and evolutionary studies. Researchers track the frequency of specific alleles, gene variants, in a population. Changes in these frequencies, genetic drift or natural selection, can provide insights into evolutionary trends. Conservation biology. Monitoring genetic diversity in endangered species for plant breeding programs. Ensuring that inbreeding is minimized by accurately identifying lineage. Medical diagnostics and transplant matching. Though more commonly associated with HLA, human leukocyte antigen, typing, DNA fingerprinting can contribute to matching donors and recipients. Helps reduce the risk of organ rejection and complications. Now, when we say DNA fingerprint, we are not talking about your actual fingerprints. We are talking about your genetic blueprint, the unique code that makes you, you. It's so unique that the chances of two people having the same DNA fingerprint are astronomically small, unless they're identical twins. These non-coding regions are full of repetitive sequences, like little broken records repeating the same phrase over and over. They're called VNTRs, or variable number tandem repeats. The keyword there is variable. The number of times these repeats happen is different for everyone. One person might have a sequence repeat 10 times, while another has it repeat 50 times. It's this difference that allows us to create a unique DNA fingerprint. The lab procedure, a step-by-step breakdown. So, how do we turn a tiny sample of DNA into a unique pattern? It's a fascinating process with a few key steps. 1. DNA isolation. First, scientists need to get the DNA out of the cells. They take a sample, from blood, a hair follicle, saliva, you name it, and use a special procedure to break open the cells and purify the DNA. 2. Digestion with restriction enzymes. Next, they use special molecular scissors called restriction enzymes. These enzymes cut the DNA at specific points, but the length of the resulting fragments will vary from person to person because of those unique VNTRs we talked about. This is where the magic begins. 3. Separation by electrophoresis. The DNA fragments are then placed in a gel, and an electric current is run through it. Think of it like a race. The smaller, lighter fragments move quickly through the gel, while the bigger, heavier ones get left behind. This separates the fragments by size, creating a series of distinct bands. 4. Hybridization with probes. Now, we need to make those bands visible. Scientists add special, labeled probes that are designed to stick only to the specific repetitive sequences they're looking for. These probes are like tiny homing beacons, lighting up the exact spots that are unique to each individual. 5. Autoradiography. Finally, they use a special film to detect the labeled probes. The result is a barcode-like pattern of bands, the DNA fingerprint. Beyond the crime scene. Now, while DNA fingerprinting is most famous for solving crimes, its applications are incredibly broad and have a huge impact on our world. It's used in paternity and maternity testing, confirming biological relationships with near-perfect accuracy. It's also a powerful tool in conservation biology, helping to track genetic diversity in endangered species to ensure their survival. Scientists even use it to study evolutionary trends by tracking changes in gene variants over time. What's the most surprising use of DNA fingerprinting you've heard about? Let me know in the comments below. Don't forget to like and subscribe for more amazing science videos. See you next time.